The issue that this article is trying to address is the fact that 100,000 patients can be found on the organ donor waiting list at any given time. Approximately 22 of those people waiting on that list will die each day. Tissue engineers have tried to come up with alternative solutions to organ failure. However, there are many issues when it comes to applying these solutions to the clinical setting. The main issue that comes across, whether the suggested method be 3D printing or another resource, is that they lack a functional vascular network. Lacking a functional vascular network does not allow for a viable graft to be created. Instead of trying to engineer these vascular networks, bioengineers have suggested bio-inspired approaches such as plant decellularization. Plant decellularization is similar to mammalian decellularization, where all of the cellular material of the tissue or organ will be removed, creating a cell scaffold, which consists solely of the extracellular matrix, which can then be recellularized with the patient's own cells. The issue with mammalian decellularization is there is inconsistency between grafts as patients vary, whether that be because of age, ethnicity, or other issues. Another issue is the high cost of this process, as well as the lack of abundance of decellularized mammalian tissue, and the lack of clinical research to support this method. Plant decellularization has instead been suggested. It is a cheaper and more consistent alternative. Plant tissue has the same vascular system as mammalian tissue, which makes this method just as viable as the others. In fact, cellulose has been studied for clinical use and is found to be biocompatible and has properties which promote wound healing and can be beneficial in the use for grafts. As we dive into the methods and results of this article, you will see that plant decellularization is an efficient alternative. This first figure compares the mammalian and plant vascular systems, and as you can see, there are many similarities in the size and shape of the vascular networks.
My name is Santoshi Kandula, and I'll be discussing the methods for this article and this experiment. The methods for this experiment are listed on the slide. As you can see, they're numbered and they follow a clear pattern. It first begins with the preparation of the plants for decellularization, and then various imaging analysis and mechanical testing analysis are applied to recording the properties of these decellularized plants. In addition, an experiment was run with embryonic stem cell-derived cardiomyocytes, or HPS-CMS cells. These cells were thawed and immediately seeded onto the decellularized plant leaves. Then the cell cultures were viewed under a fluorescent analysis, and a MATLAB program was run to measure the intensity change over time. So first I'll be detailing the first couple parts of the methods. The first part of the methods is the plant decellularization process. Plant decellularization first begins with spinach and parsley seeds that were acquired from local markets, Artemisia annua leaves, which were harvested from soil-grown plants, and peanut hairy roots, which were generated through Agrobacterium rhizogenes-mediated genetic transformation. These plant parts were decellularized using serial treatment with hexanes and deionized water solution. After this process, the tissues were stored in sterile deionized water at 4 degrees Celsius until needed. So these tissues can be stored in this water for about 2 weeks. After the decellularization process, a histological analysis was run. For this analysis, leaf samples were cut into 1 centimeter squares, making sure that the main vascular channel down the middle of the plant was still intact. These samples were fixed overnight with an ATP1 atomic tissue processor. These samples were then stained with safranin so that they're easily viewable under a fluorescent microscope or under any microscope. The sections that were cut were dehydrated in ethanol and then washed and then visualized under the DMLB2 upright microscope. The next step of the methods is a DNA and protein quantification. Both the decellularized and native leaves were centrifuged, and the DNA content was measured using a PsiQuant direct cell proliferation assay, and the protein content was measured using a Coomassie protein assay. The respective concentrations of each were determined. Some of the leaves that were run through were perfused with a 0.1% Poncio red solution. This is a dye that perfused to the cannula of the leaf, and as you can see in this slide, as I forward through the PowerPoint, the leaf is a red color, showing the perfusion of the dye through the leaf, through the decellularized leaf. Yes. And then the next step is the mechanical testing that was run. The leaves that were decellularized were loaded onto the grips of a mechanical tester and pulled for a certain amount of time. Mechanical properties were tested, including max tangent modulus, max tensile strength, and failure strain. These results were recorded and will be detailed later in the presentation. In terms of the other experiment that I mentioned, the HPS CMS cells that were adhered to the decellularized plants, many statistical tests were used, and these tests are presented in the results section of the presentation.
The results at each step of the decellularization process and recellularization process were documented. The first set of results characterized the cell scaffold both before and after the decellularization process. Histological staining showed that lignin was still present. Lignin is a major component of the leaf vasculature. It is also important to note that the vascular network is the same size and density both before and after decellularization. Additional tests were also conducted to analyze the presence of DNA and protein after decellularization. The results showed that 9.4 nanograms with a standard deviation of 1.3 nanograms of DNA and protein were present after decellularization. This is in comparison to the initial amount of 1,129 nanograms with a standard deviation of 217.3 nanograms. This indicates that nearly all DNA and protein was removed during the decellularization process, and the remaining levels satisfied the minimum requirement to classify the material as decellularized. Additionally, the mechanical properties of the decellularized tissue were analyzed. These results showed that there was a lower tensile strain and stress failure during uniaxial mechanical analysis. The modulus of this material was also found, and it was found to be 0.3 megapascals. This is within the range for normal decellularized human cardiac tissue. Further tests also show that plants aside from spinach leaves could be used for this method and perform adequately. The first figure shows a time lapse of spinach leaf decellularization over the course of the 7-day time span. As you can see, on day 0, there is a perfectly normal cellularized spinach leaf. On day 7, there is a clear decellularized spinach leaf. This next image shows a side-by-side comparison of different characteristics before and after decellularization. These next set of results were conducted on plant vasculature post-decellularization. To investigate the clinical relevance of the decellularized tissue, fluorescent microspheres at various diameters were perfused to evaluate the diameter and patency of the vessels. It was found that most spheres less than 50 micrometers in diameter were perfused sufficiently, while any cell over that diameter was seen to be less present at the end. Regardless, these results support the conclusion that the vessels can transport red blood cells. This next figure highlights the perfusion capabilities of the decellularized plant's vasculature network. Further tests were also done to investigate the human cell application of the recellularized plant tissue. First tests were done to analyze the human cell's ability to adhere to the plant's extracellular matrix. Positive fluorescent light indicated that human umbilical vein endothelial cells were viable after recellularization. However, methods still need to be improved before complete endothelialization of the entire cell can occur. Human mesenchymal stem cells were seated on the outer surface of the decellularized leaf. This cell adherence was completely observed after 24 hours. This next figure is an image of human cells being used to recellularize the plant tissue. Further tests were conducted to analyze the functionality of human pluripotent stem cell-derived cardioid myocytes. Results showed that contractile strain increased until day 10, then peaked and plateaued. A decrease in contractile strain on day 21 prompted further testing, which
Hi, I'm Abum Okemgbo, and I plan on discussing the next reasonable steps that one would take in order to further this research. First of all, this research had its limitations. With all of the aspects of the experiment done in vitro, we do not know how the body would directly respond to the implanted vascular network. Additionally, the possibility of biodegradation over time adds fears that the implanted heart would not suffice during its duration of use. Alongside these possibilities, there is the problem that although there are similarities between plants and human networks, they are not identical. Plant networks do not need for outflow vasculature characteristic in human blood vessels. Consequently, in the future, one may look for options that closely mirror blood vessels. Since we saw decreased mechanical properties after decellularization, we aim to maintain the strength of our network so it can be applied wherever in the body it is needed. Additionally, in this experiment, we saw a decrease in the flow rate after the decellularization process. While this is an acceptable flow rate for smaller particles, we begin to run into problems with larger particles. While the size of a blood vessel fits into the range of the smaller particles that I was mentioning before, we may worry about clotting when applied to the actual heart. As a result, we want to maximize the overall flow rate. I guess our overall impact, there's an impact in the environment with animals, in our medical applications, and the cost. So first of all, for the environmental aspect, a lot of materials are made with plastics which use non-renewable resources such as petroleum to make, and alongside we sometimes use Teflon in our biomaterials, which often creates toxic byproducts, which we want to avoid. And so with plants, we don't have to even deal with this. With animals, if we use animals, then we have to always kill the animal or usually kill an animal in order to take the heart or whatever organ we're using. And so with plants, we don't have to deal with any ethical issues dealing with this. And along with this, animals often have upkeep necessary to survive their use. So for medical applications, there's a variety of plants throughout the world, so that gives us a lot of options in which plant we use. So we may use this more vascular plant for something like the heart, like putting blood throughout, like capillaries. But then we may want to use a larger vascular network, like something from a tree, if we're trying to represent an aorta. Along with this, since we're using living materials, this could decrease our chances of having biocompatibility issues. Additionally, plants are easy and cheap to obtain. We saw earlier we use spinach leaves from the market, so this is relatively inexpensive. So overall, this provides us with a promising solution to our problem. Obviously, we've mentioned before that we have a huge shortage of organs needed for people who are sick, and so more research needs to be done in order to see if we can actually apply this to people. So we've looked at other alternatives, and we see that they are not near as good as this option, as in loading, as the ones listed down here. They don't deal with the aspect of having to use a vascular network. So this proves to be a good option. And so I guess thank you for listening to our presentation. And I guess that's all.